Question number 176. A new breed developed by crossing Bikaneri Eves and Merino Rams is, and this is Hessardale, which is a crossbreed. So the correct answer to this question is option number 4, Hessardale. The very first option, mule, is incorrect. It is an example of interspecific hybridization. Option number 2, Leghorn, and option number 3, Jersey, are incorrect options because Leghorn is a breed of poultry, whereas Jersey is an exotic breed of cattle. So the correct answer to this question is option number 4. Let's move on to the next question, question number 177. If genes of interest A and B are simultaneously ligated at the restriction site of PST1 and PVU2 in PBR322 cloning vector of E. coli, what will happen to the recombinant host cell? Now, if this is PBR322, has a site at ampicillin resistance site for PST1. Here you have tetracycline resistance gene, and here you have a rope which have site for PVU2. If the gene of interest is ligated at here PST1, that will lead to insertional inactivation of ampicillin gene, as well as at PVU2, that will lead to insertion inactivation of rope gene 2. They become inactive, whereas as tetracycline resistant gene is still intact, there would be resistance towards tetracycline. So in recombinants, there would be tetracycline resistance but ampicillin sensitivity. But as the gene is inserted at PVU2 site, leading to insertion inactivation of rope that is responsible for coding proteins involved in replication, so these recombinants will not be able to replicate itself in host cell. Now let's discuss the options. The very first option is it is resistant to ampicillin and will grow on XGAL containing medium. This is incorrect as the recombinants are ampicillin sensitive. Option number 2, the plasmid will not replicate inside the host cell, is the correct answer to our question. Option number 3, blue colored tetracycline resistant colonies will grow on XGAL medium. This is incorrect as there is no LAGZ gene that would create blue colored colonies on XGAL containing medium. Fourth option, white colored ampicillin resistant colonies will grow on XGAL medium, is also incorrect as there is no intact ampicillin resistance gene in the recombinant colonies. So this is an incorrect option. And the correct answer to this question is option number 2. Let's move on to the next question, which is question number 178. Which of the following is the incorrect statement during and after gel electrophoresis? The very first option is cutting and extracting the separated DNA fragments from agarose gel piece is called elution. And as it is a correct statement, this cannot be a correct answer. The next statement is the smaller the DNA fragment, the farther it moves from a node. And this is an incorrect statement, so is the correct answer to our question. Because DNA fragments being negatively charged will move towards the end node. And the smaller the DNA fragment, the farther it moves from cathode. Next statement is option number 3. DNA fragments separate according to their size through sieving effect provided by gel is a correct statement, so cannot be a correct answer. Option 4, the statement is separated DNA fragments can be visualized under UV light only after staining with ethidium bromide is also a correct statement, so cannot be a correct answer. And the correct answer to this question is option number 2. Let's move on to the next question. Question number 179. Choose the correct match. The very first match says rosy and the match is alpha antitrypsin. And this is an incorrect match, as the correct match should be rosy with alpha lactalbumin. Second match is Tracy with alpha lactalbumin is also incorrect because Tracy produces alpha antitrypsin. Third option, Hemulin, gene subtraction. This is also incorrect. And the last option is SCID, and the match given is ADA deficiency, as it is a correct match. This is a correct answer to our question. Let's move on to the next question. Question number 180, which is the last question of this AIATS paper. The question says, the term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment from the countries and people concerned is. Options are bioethics, biopiracy, biopatent, bioprospecting. And the correct answer to this question is option number 2, biopiracy. Let's discuss rest of the options. The first option, bioethics, is incorrect because bioethics may be viewed as a set of standards that may be used to regulate our activities in relation to the biological world. Option number 3, biopatent, is incorrect. Patents are supposed to be novel, non-obvious, and of utility for human welfare. Option number 4, bioprospecting, is incorrect. Bioprospecting refers to exploring molecular genetics and species level diversity for products of economic importance. So the correct answer to this question is option number 2. So with this question, I would like to end up the discussion of this AIATS paper. I hope all your doubts and queries have been solved regarding this paper. Thank you very much for watching and all the best.